test, starch test, we discussed. No, check. Got your books. So we must have completed the concept of photosynthesis. If we are not done with the starch test, then that is the last topic there. We are talking about photosynthesis. We are talking about photosynthesis, and now we already understood what are the basic phases of photosynthesis. Anything to clarify there? Any concept that is not understood? We discussed what we came across in that topic, the basic concept of autotrophic nutrition in plants. We came across photosynthesis, equation, to write it down correctly, very important, you should know that. What are the basic concepts of the raw material you should know? I have discussed under certain heads, that is how you should remember. Apart from that, the raw material, yes.
I have doubted, ma'am. There is a condition means in opening and closing of stomata.
Question that was.
No, not a question. The concept.
Process.
Yes.
Process I haven't discussed it, I suppose. Stomata mechanism, I don't think so I have done with it. But
Yes.
check for endosmosis and all these things. Did I discuss already?
Yes.
Stomata. Did I discuss with you?
Ma'am, you've done the cross section of a leaf.
Did you draw the section of the stomata? Section of stomata you haven't drawn. No, that means I have not discussed that already. Because if you are saying event, then I must not have discussed that with you. That is up. Wait. That was the last section. So now, when we are talking about what did we learn, let me just summarize. We have already talked about the processes, that is the light and the dark phases. For the light and the dark phases, let me again tell you, if you are referring to any other books apart from NCERT, you may come across various reactions there. These reactions are not there for your syllabus. You are only to understand the basic phases. Now, go to that point that I already told you. The three phases, the three events. So that is the summary that we discussed. The events of photosynthesis. Are those 3 points clear? And now when I talk about the dark and the light phase of photosynthesis that we have studied before that, what is the light and the dark phase? Under the light phase, what is the basic concept? We are talking about certain reactions which take place in the granular. Reactions are dependent on sunlight. One of the most important reactions is the breakdown of the water. Only that much. The other side, dark reaction takes place in the stroma, not directly dependent on the light. And one of the most important reaction is the reduction of carbon dioxide to form carbohydrate. Did we study that way? I think the other day we had already finished all these topics, the basic concept of the dark and the light phase. And then we were summarizing that after we know about the chlorophyll, the chloroplast, after we know about the oxygen and the glucose, what happens to the glucose and the concept of the sucrose and the starch and the translocation and all these things we had already separately studied one after the other. And next we came to the concept that if photosynthesis has to be summarized, then in how many steps we summarize? The first one was absorption of chlorophyll by the light energy. Absorption of light energy by chlorophyll. Second is the conversion of the light energy to chemical energy. And the splitting of the water molecule to give hydrogen ions and oxygen. And there again, I had already asked you to remember another concept, that how is the oxygen release? From where is the oxygen release? During photosynthesis, which is a common question we will get along with that. And the last step is the reduction of the carbon dioxide to form carbohydrate. What is the form of carbohydrate? Always to remember glucose. What is the stored form of glucose? Starch. What is the form in which the organic food is distributed? Sucrose. So all these questions, remember. So this is what we are done. Now next, after you know now the events, we have to talk about another concept of photosynthesis, that is, we will be talking about the ideal test now. What do we do here? What are we trying to test? We are trying to test the amount of starch that is present in a leaf. Starch that is present in a leaf. How do we do this? We are supposed to take a potted plant. We will take a potted plant with well-developed leaves under soil, well watered. So this is the condition that we take. A potted plant, well watered, well developed roots, soil well watered, well developed leaves. Now what do we do? This particular plant has to go for destarching. What is destarching? What is the meaning of destarching? Allow the starch to be utilized. Whatever amount of starch the leaves have, allow the starch to be all utilized. How will this be done? By keeping the plant in dark for minimum 24 to 48 hours. It can be kept for 3 days also. So, for a period of 24 to 48 hours, keep the plant in dark. Keep the plant in dark means, am I also talking about that keep the plant in any room where natural light is there? Artificial light is there? No, no natural light also, no artificial light also. Absolute darkness. The plant should be kept in absolute darkness for 2 to 3 days, 4 days, and after that, we are very much sure that whatever amount of starch was there in this leaves, it is all used up. It is all used up. Now, depending what is the shoot, what is the length and height of the plant that we are talking about, we may keep for minimum 2 days also. Then what? Then we take this potted plant and again we are supposed to keep it under sunlight for at least a period of 6 to 7 hours. At least for a period of 6 to 7 hours, keep it under sunlight. We have destarched it. You know now what is the meaning of destarching. What is destarching? Keep the plant in dark and allow the starch to be all used up. Then what to do with that plant? Keep the plant again under sunlight minimum for a period of 6 to 8 hours. Why now? Why keep the plant in sunlight? Keep the plant in sunlight so that new glucose molecules are formed in the leaf. And in this period of some 6 to 8 hours, the glucose that will be formed, that will again get converted into starch. And we are going to go for certain steps to understand how much of the starch the leaves have prepared. Now next what we do, we go selecting certain leaves. We have to take certain leaves, and with these leaves we go for a certain number of steps. What is the first step? Boil the leaf in water. The first step is boil the leaf in water. What is the necessity to boil the leaf in water? Kill the cells. That is what most of the students always write. Kill the germs. We have nothing to do with microbes here. Kill the cells. Why kill the cells? Because now that I am doing the experiment with this leaf, the leaf cells still have chlorophyll. The leaf cells still getting sunlight. The leaf cells are still having adequate amount of water. During the course of my experiment, the process of photosynthesis should not continue. Right from the time I have selected the leaf, I have to go through certain processes where the process of photosynthesis is strong. Then only I am able to measure and find out how much of the starch is collected here in this leaf. So we pluck the leaves, and the first step is boil the leaf in water. You will be asked reason, why do we boil the leaf in water? What is your answer? Kill the cells and not allow the process of photosynthesis to continue during the course of the experiment. Next step is boil in alcohol. Next step is boil the leaf in alcohol. Now that we are talking about boil the leaf in alcohol, now why boil the leaf in alcohol? The moment we start boiling the leaf in alcohol, take it in a test tube or a beaker and boil the leaf in alcohol, what will happen? The entire alcohol immediately turns green in color. And what happens to the leaf now? Does the leaf now show the same green color? No, the leaf is now faded out. The leaf is now faded out. The leaf is now faded green in color. Faded green, light green in color now. Maybe earlier it was quite dark. So what has happened after the process of boiling in alcohol? All the chlorophyll got dissolved in alcohol. Again, why are we trying to remove the alcohol? So that Now that we want to try testing with certain chemicals, we are able to see the color change. If the medium is darker, if the color is green, dark green, will we be able to see any faint change in the color? If it's all happening, we will not be able to see. That's why remove the chlorophyll. That's why remove the chlorophyll and now the change in the color. Now next is what? Rinse in water. Rinse in cold water. Now why rinse in cold water? To wash away all the alcohol so that what type of chemical droplets we are going to add now? The iodine especially that we are going to add now. The chlorophyll and the alcohol and the iodine should not enter into any such type of a chemical reaction. Wash, rinse the leaf in water. Why rinsing? Rinsing because to wash away all the alcohol. To wash away all the alcohol so that the leaf does not enter into any new type of a chemical reaction and a new type of a compound and a new type of a color develops. No, that should not happen. Yes. If you are rinsing in hot water, the leaf will start becoming wrinkling now. It will start getting shrunken now. We don't want the shrinking of the leaf right now. If we are talking about boiling it in hot water again right after alcohol, as it is, alcohol makes the leaf little bit harder. And now you put in hot water, it will start getting shrunken and performing the experiment with this iodine molecules with the iodine drops will be very difficult. So we want the leaf thin, spread out, faded in color, so that we are able to see any type of a chemical change if possible. Now what is the fourth step? The fourth step is put iodine drops. What is it? We put some iodine drops. Now what is the basic color of iodine? The basic color of iodine I should say is yellowish or say yellowish orange. A light tinge of orange is there. It's dark yellow. Yellowish orange is the actual color of the iodine solution. But the moment you are putting this iodine drops here, the moment you put this iodine drops here, what will happen? There will be the iodine drops spread out and certain patches immediately gets formed. Certain patches immediately gets formed. But are these patches very, very dark and remarkable? No, they are not very dark and remarkable, but you can understand, distinguish it there. Then, what are these patches? Two types of patches you will come across. One is blue-black in color, dark bluish in color, and the other would be brown in color. So, the entire leaf we can come across the moment we have spread out the iodine drops. The leaf now shows two distinct regions. Certain region bluish black and certain region brownish. What is the indication? Now what is the interpretation? When tested on this or when further tests are done on this blue-black regions of the leaf, it is understood that these are the cells which are storing the glucose in the form of starch. So when we talk about the blue-black region, what is the observation? The blue-black regions of the leaf indicate the presence of starch, and when the cells of this particular region are tested, it was found that they don't contain starch at all. Is this experiment understood? What are the steps and why the steps?
Only the A1.
The first step I told you is boil the leaf in water. Why boil the leaf in water? To kill the cells, not to kill the microbes at all.
Why to kill this?
What I told you? What I told you?
The photosynthesis process
Will continue. I am not killing the cells, and as I lay down the leaf and continue with the rest of the steps, alcohol will not kill the cells. Alcohol will at the most only dry up, absorb little bit of water. Only that much alcohol will do. But I want the cells to be all dead. The chloroplast part should be destroyed. Why? We have to boil it, because if I am not boiling it, the cells are still alive. The cytoplasm part and the plasma membrane cell wall part is all alive, and the cells may still continue the process of photosynthesis. So whatever, for an example, if I understand that this entire leaf has 100 gram of starch, during the course of the experiment another 50 gram may be produced. No, because this entire experiment will take another 1 to 2 hours. So So in this entire process, by following the steps one after the other, another 1 hour goes, another 1.5 hour goes, maybe another 50 gram of starch is already produced. But I don't want to measure this. I want to measure what is manufactured here at this time. So इसमें discrepancy आ गया ना. So that's the reason we have to kill the cells and stop the process of photosynthesis and then go on.
This
What?
mainly to prevent the photosynthesis.
Prevent the process of photosynthesis during the course of the experiment. Prevent the course of photosynthesis, and that is always applicable for any type of an experiment we do. When we are doing that experiment, at that time, the processes should not happen. Stop the process and then check whatever parameter you want to check. Is that clear, all of you? Now, in your exam, what type of questions you get? You get diagram-based questions like this. Application questions you will get majorly from this. Case study based questions, assertion questions you will get from this. Various different types. So the understanding of the concept is very much necessary. It's a simple concept, but try understanding because assertion questions also you get on this. So basically what I am talking about is why iodine drops to put. We are testing starch. You get questions. The iodine test, with the help of iodine test, we can test. With the help of iodine test, we can estimate the amount of the starch and not the glucose. Why? Give reason. What to write? With the iodine test, can you tell me, with the iodine test, if you are estimating starch, that is the entire story I am telling you right now. But why am I not mentioning that we can also estimate the amount of the glucose? Why can't we estimate the amount of the glucose?
And because we don't know the exact amount, how it will break down.
No, I gave you the answer already in the previous
So
part.
what is the question?
I asked you. Iodine test, with the help of iodine test, you can test starch. But photosynthesis prepares glucose, no?
Starch is the stored form.
Stored form. And what else to mention in your answer? My question was, iodine test cannot estimate the amount of glucose but the amount of starch. Give reason. What will you try? Glucose is immediately converted to sucrose and then starch. Glucose is not a stable form that remains in the plant cell for a long time, but starch remains in the plant cell for a long time. That's why we can test starch. Glucose is just a transitory state. It's just a temporary state and immediately gets converted into starch. Is this clear? So this is about the concept of the iodine test. You have a set of diagrams in your textbook also. Go through those set of diagrams. Those of you have the textbook, you can tell me what page is it? All of you, yes.
And we boil the leaf in alcohol just to remove the chlorophyll.
I gave you those conditions also down there. So whenever why we are boiling in water, why we are boiling in chlorophyll, in alcohol to remove the chlorophyll, why we are rinsing them, etc. Now this is all about the concept of photosynthesis, and the concept of photosynthesis also involves what? The concept of photosynthesis also involves the stomata, a basic structure which is very much involved. Now we were talking about photosynthesis. I want to find out what is the sectional view. Similar diagram I had explained you the other day also when I was explaining you the concept of chloroplast, chlorophyll. Same concept. I again apply. The same concept again I am showing you a similar diagram you have in your book. Now this entire section of the leaf that I understand, this section of the leaf has various layers of cells. Try understanding this. This entire leaf that we understand undergoing the process of photosynthesis, this will have a specific layer of cell, and this specific layer of cells, what is this layer of cells known as? Upper epidermis. Yes, this is the upper epidermis. What about this? I will write. This is the lower epidermis. What am I talking about? I am talking about the section of the leaf.
Can you please repeat what happens when we put iodine drops in the...
What I told you is when I put iodine drops on the leaf, the iodine drops as it spreads over, certain chemical reactions happen. And in just 5-6 minutes, we get to see some color change on the surface of the leaf. patches, certain patches of blue-black color and certain patches of brown color. Meaning all over the leaf, what happens? The entire leaf is light green now. The moment the iodine spreads, certain patches of blue-black. Surrounding regions which were faint green, that also start getting converted into some light brownish color. So now what does it show? Suno, you are now in the 10th standard. Focus. Suno, what the teachers are explaining you, understand very nicely. I already explained you this. And twice मैंने बोला है already. What I told you is, the moment the iodine drops spread out, the cells have got certain type of a chemical component. Every cell have got certain type of a chemical component in them. The iodine drop spreads. Wherever starch is there, this group of cells will start showing a blue-black color. Maybe this group of cells not undergoing photosynthesis. I already told you at the beginning also, though it is a leaf, every cell is not showing photosynthesis. These cells are showing photosynthesis. These group of cells not showing photosynthesis. So what will happen? This group of cells will show a blue-black color and this group of cells will not show a blue-black color. So that is what we get to see. Certain patches of blue-black color and the surrounding regions are all brown. Now, when tested, what is being found? That these are the cells where the component known as starch is there. These are the cells, starch absent. Similarly, if I have to take, say suppose a starch solution, a simple starch solution. A simple starch solution, I take a test tube, this much of water, put some 1 teaspoon of some atta or maida. These are all big sources of, very, very common sources of starch. Put some corn flour, put some wheat flour, whatever. Take some 1 spoon of the wheat flour or the corn flour in this test tube. Mix it. Put 2 drops of iodine. The entire solution immediately will turn blue-black. So what is the indication? The indication is, what is the common reaction? The most common reaction that we are talking about is, iodine when reacts with starch always shows a blue-black color. Iodine when reacts with starch always shows a blue-black color. Now what I am telling you is that in the entire process of photosynthesis, stomata plays an important role. Where is the stomata present? So that's why I have drawn this diagram. We again try to look into the section of the leaf. Now, in the section of the leaf, we come across, in the lower epidermis, there is a pair of some bean-shaped cells, and these bean-shaped cells are known as the guard cells. We first of all try to understand that what is the function of these guard cells. What is the function? What are the 2 most common functions of the stomata that you should know? What are the 2 most common functions of stomata? Release of water. So if this is the stomata, we now majorly try understanding with respect to photosynthesis. This is the stomata. Now why am I drawing the stomata here? Why not here? Because maximally present on the lower surface. For any particular leaf, for any particular dicot leaf, if I talk about, is it that stomata not present here? No, here also they are present. But if here on the outer surface, on the upper surface, I come across in one small space, I come across that 10 of them present, maybe now here in the same area, 50 of them are present. So what is the difference? Always on the upper surface, on the upper epidermis, the number of stomata is much lesser. Lower epidermis, the number of stomata numerous. Why is that? Why we have eyes here? Why not here? That's the adaptive characteristics. That is the adaptive. These are all called adaptive characteristics. So when we are talking about this particular leaf, these are just the characteristics of the leaf to adapt with the environmental conditions. You talk about a desert leaf, absolutely different feature. You talk about a leaf in the water, absolutely different feature. Most of the leaves on the land plants that we understand, they will always have this type of a feature so that they can take in a large amount of carbon dioxide, give away oxygen and the transfer of the water. Now with reference to photosynthesis, I have to understand that what is this guard cell's role here? What is the job of the stomata here? Two functions, always you are asked to write. What are the two functions? Intake of carbon dioxide and the giving out of oxygen. This is the first function you will write. Exchange of gases, inward movement of carbon dioxide and the outward movement of the oxygen. And apart from that, you also understand the release of the water in the form of vapor. That is the concept of transpiration that we understand. Now, right now, we are not talking about the concept of transpiration because we are talking in relation to photosynthesis. Now, let us try finding that how is this stomata opening and closing to bring about the process of photosynthesis. To understand this, also try understanding that this is a stomata. Surrounding the stomata, there are some various group of cells. And there is another arrangement of the cells, another type of a tissue. I think all of you remember. What are those tissues which help in the conduction of the water?
Xylem.
Xylem. Is the xylem there in the root? In the stem?
Yes.
In the branches?
Yes.
In the veins of the leaf? Yes, and in the leaf cells also. So can I say that if I talk about a group of cells here, say suppose, surrounding the guard cells here exactly, there are another group of cells, and these group of cells I have to also take into consideration that in different regions of the leaf, there are these type of cells also present, known as the xylem cells. And these are in the form of a tube, no, connecting the root to the upper parts of the plant. So this concept also to remember that why suddenly I am drawing a xylem here, because right from the root and the stem and the branches and the etc. to reach the leaf, the xylem is almost in the form of a tube connecting every part of the plant right from the root. Clear? Am I clear? The xylem tissues that I am talking about is almost in the form of a tube connecting the root to the upper parts of the plant. So with that relation, I am drawing these xylem cells close to the guard cells. And also now here you are supposed to understand that these xylem cells will give the water to these cells, the xylem cells will give the water to these cells, the xylem cells will give the water to these cells. Understanding? So now when I talk about this group of xylem cells, are these xylem cells present in different regions of the leaf? Yes. The xylem cells are very much present in a specific region of the leaf. How will these guard cells and the surrounding cells get the water from the xylem? Now, if that is true, then we have to come to understand here that for the process of photosynthesis, what is the role of the stomata? For the process of photosynthesis, should the stomata remain open for a long period of time? Yes. Q, because the process of photosynthesis requires carbon dioxide. Higher the absorption and the diffusion of carbon dioxide, higher is the rate of photosynthesis in these cells. What is the name of this type of cells? Mesophyll cells. So this carbon dioxide has to enter this way and then keep on diffusing and diffusing to reach this group of cells, and that is how this group of cells undergo the process of photosynthesis. Clear? Now, if that is true, we have to now understand that how are these guard cells opening and why do they close? So here I talk about guard cell. Now these are the bean-shaped cells, and these bean-shaped cells, what type of a feature do they have? The outer wall, this outer wall that I draw, this outer wall is thin. This is a plant cell. This outer wall is thick and the inner wall is thick here. Watch it. This is a plant cell. It should definitely have the vacuole. This is a plant cell. It should definitely have the vacuole. What else? This is a plant cell. It should also have certain cell organelle. Which cell organelle we are talking about? Chloroplast. And on top, right? What happened? On the yellow and the red and the blue, concentrate, focus on the topic. Now, when we are talking about the guard cells, are there any surrounding cells? Yes. There are names to these cells. Look at the diagram that you have in your volume. Mark this diagram, you will have to draw. Just keep mark those diagrams, the open and the closed stomata. You will draw it as homework, not now. Just mark and keep, and now you try understanding the process. So now, what is happening is we are talking just mark and keep, and then concentrate, understand the terms and the process here. So now, when we are talking about this structure of the stomata, what is happening? We are trying to understand how does the stomata open and close. And it is very much understood that the stomata opens during the daytime, remains open for a longer stretch of the day so that the diffusion of the carbon dioxide is adequate to bring about a higher rate of photosynthesis. But then how? Here I say that if these are the xylem cells nearby, and the xylem cells are already filled with water, the xylem cell is giving water to this cell, the xylem cell is giving water to this cells. Let us start from the concept of chloroplast to understand the theory here. We start from the concept of chloroplast. What are these? Remember, I am talking about the cell organelle. Which cell organelle? The chloroplast. The chloroplast is a cell organelle. Already you know this. Where is the chloroplast? Present in the plant cells. What is the job of the chloroplast? What is the job of the chloroplast? What is the chloroplast doing? What is the chloroplast job? Already we had understood with the concept of photosynthesis. What will the chloroplast do? What is it doing? Traps. Chloroplast traps sunlight. No. Chloroplast does not trap sunlight. What is the chloroplast doing? What is the chloroplast doing? No. What is the chloroplast doing? What is the function of chloroplast? Forms chlorophyll. Manufactures chlorophyll. What is the function of chloroplast? You concentrate. You are not concentrating at all. You are smiling away and dreaming away something. I'll have to pull you out of the classroom and ask the reason. Once you are inside this classroom, let me make it very clear to you all children. You are inside this classroom, everybody cannot afford this place. Very sure. Everybody cannot afford this place. If you are here, see that you learn. Once you listen to other teachers, etc., we all different subjects are teaching, then CBSE is nothing to crack. We believe everybody can easily get 100 on 100 on each of those subjects. You don't have any idea how much this 10th ICSE children are getting. How much? More than double your syllabus. You all are getting 100 on 100 on those subjects. And 10th CBSE, you all are struggling. What you all are studying is 6th standard of their level. If you all are aware. Concentrate करो, otherwise ये simple concept 4 बार बोलने की जरूरत ही नहीं है. एक बार बोलेंगे तो समझ में आ जाता है समझ. Now, when we are talking about the chloroplast, learn one by one what is happening. We start from the chloroplast. What is the job of chloroplast? Produce chlorophyll. Yes. Chloroplast produces chlorophyll, and chlorophyll will trap the solar light, solar energy, and as a result, the process of photosynthesis happens. So, as a result of photosynthesis, what is the product of photosynthesis? You know now already. The product of photosynthesis is glucose. So now let me understand that this particular vacuole of the guard cell which has sap. You know what is sap? I already used this word earlier. What is the sap? Sap means नहीं. What is sap? Plant cell में sap होता है. Last time also you have studied no plant cell. Plant cell में sap होता है. Sap का क्या मतलब है? Cell sap पढ़ा तो था. That I also know. What is cell sap? Disgrace. Cell sap भूल गए? That one also was there, no? Cell sap. What is cell sap? The vacuole of the plant cells. I talk about a guard cell now. Every plant cell has got the vacuole. The vacuole is filled with water and minerals. So what is there inside? Water and minerals. So the water and minerals of the plant cell is known as the sap. Remember that. So what is sap? Inside the plant cell, the amount of the water and the minerals. The water and the minerals. Now I am trying to understand that this glucose is getting dissolved in the sap. The glucose is getting dissolved in the sap. As the glucose is getting dissolved in the sap is getting concentrated. So I would say the sap is becoming hypertonic. Are you understanding? I told you I am talking about only this guard cell now. Am I talking about the surrounding cell undergoing photosynthesis? Is this cell undergoing photosynthesis? No, because इसके पास chloroplast नहीं है. So now this particular cell which is undergoing the process of photosynthesis because it has chloroplast, chlorophyll release and absorption of sunlight, everything, photosynthesis is happening, and now it is forming the glucose. This glucose is getting dissolved in the sap, making the sap concentrated. Then whatever is the sap here, this sap is concentrated. Then whatever the sap here, this sap is concentrated. Then because ये भी plant cell है, sap तो इसके अंदर भी है, but this sap is not getting that concentrated like this cell. क्यों? क्योंकि these cells are constantly undergoing glucose formation and sap concentrated हो रहा है. As compared, this has got more water. This is becoming concentrated. Are you understanding? Right, remember the concept of hypertonic, hypotonic, all these things that we have studied. Now, if the sap is hypertonic, I am saying, regularly glucose is getting dissolved, the cell is becoming hypertonic. So, अगर ये है, then is it not? The cell will start absorbing water this way. Will this happen? This particular cell is concentrated, this is dilute, this is dilute, this is concentrated, this is dilute, this is concentrated, this is dilute, this is concentrated. So what is the direction of the water movement? This way, no? So what is the direction of the water movement this way? Now, if I understand that this particular cell is absorbing water, the cell is absorbing water, what is that type of osmosis? Endoosmosis. So what is happening? The guard cell is undergoing endoosmosis and absorbing a lot of water from the surrounding cells. It is absorbing a lot of water from the surrounding cells. It is absorbing a lot of water from the surrounding cells. Again, I repeat. The surrounding cells, क्यों dilute है as compared to the guard cell? Because the surrounding cells are not manufacturing glucose. इसमें photosynthesis नहीं हो रहा है because they don't have chloroplast. Now, if that is true, constantly by absorbing the water, the vacuole is becoming filled with water. So the vacuole is becoming distended, tight filled with water and extended. The vacuole is swelling up. Yes, the vacuole is swelling up. The vacuole is becoming filled with water. As the vacuole is becoming filled with water, now next what? As the vacuole becomes filled with water, this the outer membrane will start extending and the inner membrane will be pushed like this. The outer membrane extending and the inner membrane pushed like this. Same here, the outer membrane extending and the inner membrane pushed. This is what is constantly happening for the guard cells. So this would be the characteristics of the guard cells now. This would be the characteristics of the guard cells now. The guard cells are now tight filled with water. I'm just giving you this alternative term. It's optional for you. If you can remember to write, good. If you don't remember, no need. The guard cells become turgid. The guard cells now become turgid. What happens to the guard cells? They are tight filled with water. What happens to the guard cells? They are filled with water. What is that word we always use? That is what is called turgid. Any cell which is tight filled with water. But now we are also trying to understand that why do we consider this condition as open stomata, because now the space between the guard cells is even more and the diffusion of the carbon dioxide is even more, like as it was earlier. Maybe at the start of the day. And now after 2-3 hours when the intensity of sunlight is quite high, the process is something like this. But the question is still what time this will continue like this? As long as the intensity of sunlight can favor this process. By 3 o'clock, 4 o'clock, as long as the intensity of sunlight can favor this process, the condition of the stomata is like this, absorbing, taking in adequate amount of carbon dioxide, and the process is going on. But now, as of now, 6:30, 7 o'clock, is the condition of the stomata like this? No. Why 6:30, 7 o'clock? What has happened? The chloroplast is not producing adequate amount of chlorophyll. There's a drop now. The chloroplast is not able to manufacture adequate amount of chlorophyll. The chlorophyll, since the deficiency of chlorophyll is there, the process of photosynthesis is slowing down. If the process of photosynthesis is slowing down, the production of the glucose is now becoming lesser. As the process of glucose formation is becoming lesser, so this glucose concentration is now decreasing in the guard cells. New glucose molecules are weak. पिछले 1-2 घंटे से new glucose molecules are not getting formed. जो glucose पहले बना था, that is already getting distributed, etc., etc. So now the sap is still hypertonic. Then now the sap is hypotonic. Now the sap has become hypotonic. Now the glucose concentration is now no more been added. New glucose molecules are not getting formed that faster. So this sap is now becoming hypotonic, and after a particular period of time, since during the daytime it has absorbed so much of water and now the glucose concentration has dropped, after a particular period of time, now maybe by 7:30, 8 o'clock, what will be the situation? This is dilute than this. Because this cell did not take up so much of water. This was greedily absorbing water to balance the concentration. ये cell, this surrounding cells, थोड़ा water they were absorbing, but they were not absorbing so greedily to balance their concentration. But these guard cells were doing so. So after a particular period of time, what happens? Because of the hypotonicity, what will happen? Yes, this vacuole, the vacuole will shrink down. The vacuole will start shrinking. Why is the vacuole shrinking now? Because now they are not undergoing the process of endoosmosis, but rather they are undergoing the process of exosmosis. What is the meaning of that? Now the water movement is this way. Now the water movement is not this way. Now the water movement is this way. Following or not? Now if the water movement is this way, then what will be the condition of the cell? The condition of the cell is becoming shrunken. The cell will become shrunken like this. The cell will become shrunken like this. Initially we were talking about extension, and now no more extension. Earlier the guard cell was pushing the neighboring cells. Now that is not happening. Your parents have sent you here, spending so much of money to share a bond and develop the love and the affection between both of you, right? 10 standard, so called. I don't see any majority of the 10 standard children here. Should I tell this to your parents? Show me your face. Better you be in love with this board now. This boy, I've been noticing you every time, even from the 9th standard. Better sit at home and study if you have to do this. You are coming here. Parents are taking so much of effort to make you sit here so that you learn something better. And you are doing nonsense in the classroom. Don't dare not to do all these things here. Nobody will allow you to sit near in that cozy corner. You'll have to sit alone. The first bench this side, or I think better that side. You'll remain straight that side. Now, remember 10th standard classroom has to be very serious. There should be enough interaction. No nonsense at all I'm going to tolerate in the classroom. You ask 100 questions, I'm ready to answer, but any nonsense you can throw you out of the class. Now, what I'm talking about is by the night time, late in the evening and by night, what is happening? This guard cells now become shrunken. And the space between the guard cells, is it wider as before? No, they narrow down. Now we are going to term this as closed stomata. What do we consider this as closed? Now remember, whenever we are talking about closed stomata, it's never that the membranes are just closed in contact. Never. At the most, little bit more than this I could have drawn, where the two membranes come in close contact almost, but they never get fused. Closed stomata, the concept of diagram that you draw, take a note. When I draw the diagram of the closed stomata, should I ever draw the diagram something like this? Can I ever draw the diagram something like this where the membranes are in close contact? Never, that is wrong. You should never draw diagrams like this. You will never draw diagrams like this. Membranes are not in close contact. So that is the concept of the closed stomata and the open stomata. What did we learn? In the concept of photosynthesis, does stomata play an important role? Very much. The stomata open for a longer period of the day. Inward diffusion of carbon dioxide is more. The rate of photosynthesis is more. By the evening, by late in the evening and at night, the stomata remains closed. When will the stomata open again? Morning, when the intensity of sunlight is more. Morning again, intensity of sunlight is more. When I talk about a plant now at night, some flash of light is falling on that plant, strong flash of light falling on that plant, will it make a difference in the opening up of the stomata? Yes, to some extent. To some extent.
Same happens in the desert or different places.
In the desert, number one, the placements of the stomata are absolutely different. If I would have been drawing a desert plant, then I will have to draw the stomata here. I will draw the stomata here, not here. For a desert plant, the stomata are sunken like this. Sunken stomata means they are hidden in between the layers of the cell. That is one of the most important features. Because they need to conserve the water. They will never be on the surface to give away a whole lot of water.
Yes.
And many more features.
At night they do the process of
Taking it. So these are some modifications that we are talking about, and with reference to opening, closing of the stomata. Homework, you will be drawing the diagram very neat and nice. Opening and closing of the stomata. Stomata diagram that you draw. If this is your notebook, instructions you know, otherwise I make you draw again. This is your notebook page. How will you draw the diagram? Open stomata in something around 8-9 lines. Closed stomata below that. Not open and closed stomata so congested and the writing and the letters and nothing is understood. Not that way. In the exam, we will not get both the diagrams to draw. We will either get open to draw the closed to draw. So draw the open stomata nice and neat and well labeled. 8-9 lines I am telling you, not smaller than that. And the closed stomata below that. Label all the parts properly. What is it? Last class that I gave you the homework. What were those two questions I gave you? What was that question?
Chloroplast and chlorophyll.
Difference between chloroplast and chlorophyll. What did you write? Wait, one at a time. Which one?
Chlorophyll. The green coloration in plant is
The green
coloration.
color.
Color.
Color, not coloration. Coloration is no word in the dictionary. Color. The green color. Then
Having chloroplast root giving the site of photosynthesis.
chloroplast. Site of photosynthesis. Go ahead.
Second, chlorophylls are present in the chloroplast.
The first point for chlorophyll, what you wrote?
It is a photosynthetic pigment.
And the first point for chloroplast, what you wrote?
It is the site of photosynthesis.
All of you check your answers that you write. When you are writing a difference, let me tell you, the parameter that you choose has to be on both the sides. For chlorophyll, site of, it is a photosynthetic pigment. For chlorophyll, photosynthetic pigment, and chloroplast, site of photosynthesis. No, it is not a comparison at all. Some of you are understanding this. It is not a comparison at all. तो अगर क्लोरोफिल को फोटोसिंथेटिक पिगमेंट लिखेंगे तो क्लोरोप्लास्ट को क्या लिखेंगे?
Cell organelle.
नहीं, you can write a genuine concept there. Cell organelle लिखेंगे. Understood what I am telling you? When you write the difference, दोनों तरफ same parameter पे लिखना है. Chlorophyll is what? Chloroplast is what? Chlorophyll का structure क्या है? Chloroplast का structure क्या है? Structure we have not studied. So chlorophyll का function क्या है? Chloroplast का function. This is what I asked you to write down. All of you check your answer. If you have written something like this, please correct.
Ma'am, can we write that chloroplast have their own DNA and chlorophyll don't?
No.
Yes, ma'am, because chlorophyll don't have their own DNA and
When you-
chloroplast have their DNA.
That I know, my dear.
That can write.
No, you cannot. Because when you are talking about the chloroplast, you are writing one feature which the chlorophyll has no relation to it. How can you compare that way? You cannot compare that way. That is what I am telling you. Comparison has to be on the same parameter. You can say that mitochondria has its own DNA and chloroplast also has got its circular DNA. That is a comparison. You cannot write chlorophyll, chloroplast, different parameters. Now, what are the 2 points that you will write? Number 1, chlorophyll is a pigment, chloroplast is a cell organelle.
Yes.
Chlorophyll traps solar energy or sunlight and function. So, I am talking about the function. Chloroplast
Produces chlorophyll.
produces chlorophyll. What is chlorophyll doing? What is chloroplast doing? Something that chloroplast does not have, and I cannot write that chlorophyll does not have and chloroplast have this. This is actually not true differentiation to write. Please try to understand.
Can we write a differentiation means in which it is present and in which it is not present?
Meaning,
Means if we take chloroplast, it is chloroplast is present in plants, algae, cyanobacteria, and chlorophyll is yes, that's sorry. And chlorophyll is present in plants and algae. We can write it. It is
chloroplast
present.
is not present in cyanobacteria.
It is present in cyanobacteria. Chloroplast is present in plant, algae, and cyanobacteria, and chlorophyll is present in plants and algae. We can write
So
the
chlorophyll अगर present है, chloroplast present कैसे नहीं है?
Can we write the in which it is present?
You are highly confused. तुम कौन सा guidebook पढ़ रहे हो? कोई भी clear your concept. What are you writing is you are saying algae में chlorophyll है, chloroplast नहीं है.
No, मैं नहीं की बात नहीं कर.
अच्छा एक और बार पढ़ो उसको क्लियर करो concept. You are writing absolutely wrong.
Chloroplast it is present in all plants, algae, and cyanobacteria.
Okay.
Chlorophyll present in all plants and algae.
जहाँ पे chlorophyll है वहाँ पे chloroplast भी है.
No one is saying that chloroplast is present.
Cyanobacteria में नहीं है.
नहीं
वही हो रहा है ना.
cyanobacteria में है लेकिन
Chlorophyll
chlorophyll नहीं है cyanobacteria.
तो वही तो मैं बोल रही हूँ. जहाँ पे chloroplast है वहाँ पे जैसे तुम्हारा सिर, जहाँ पे तुम जा रहे हो तुम्हारा सिर भी तो जाएगा। Exactly that one. क्लोरोप्लास्ट और क्लोरोफिल अलग कुछ है ही नहीं ना। क्लोरोप्लास्ट एंड क्लोरोफिल ऑलवेज टुगेदर। वो पॉइंट अगर तुमने कहीं से लिखा है करेक्ट पढ़ो, वहाँ पे इट इज नॉट लाइक क्लोरोप्लास्ट एंड क्लोरोफिल प्रेजेंट इन ऑल डिफरेंट टाइप ऑफ प्लांट सेल्स, certain type of algae, cyanobacteria, etc., etc. That's a common point for the chloroplast and chlorophyll both. Understood? Now, you will complete the diagram. The next class we are going to start with nutrition in animals. We are already done with nutrition in plants. We will start with nutrition in animals. Read and come the textbook also this part. If there is any doubt, you will have to clarify from time to time.
washing machine cycle भाई ये क्या था, ये क्या है? वॉशिंग मशीन आरसीबी हो गया, आरसीबी हो गया। साइकिल का वॉशिंग मशीन के साइकिल, अरे वॉशिंग मशीन के साइकिल एक वॉश वो खत्म हुआ,
छूट जाना चाहिए, ठीक हो गया है हमारे बार टूटना है
इसका पार्ट आज आस्ता आस्ता वो भी नहीं सुनी मेरी सिल्विया का लगता है कंसंट्रेशन आ रहा
बंद कर चल
थोड़ा सा बाहर पड़े लोग
भाई चल भाई चल